Hello. So these are some MCQs of post-graduation of any entrance exam, and also about of PhD entrances. This is the first question, part A, that is of general aptitude, that is of maths. The first question is 36 workers can finish a piece of work in 14 days. If the work is to be completed in 8 days, how many extra workers are required? So 36 workers is M1, 14 days is T1, that is time 1. If the work is to be completed in 8 days, days means time, so how many M2 extra workers are required? So here, when work is, this is about time and work, this is from chapter time and work. So the concept applied is when work is same, M1 T1 is equal to M2 T2. So M1 is 36, time 1 T1 is 14, M2 we need to find out, T2 is 8 days. So it will go here, M2 comes out to be 63. So M2 comes out to be 63. Now here they are asking about extra workers. So extra workers, that is 63 minus 36, that is M2 minus M1. This comes out to be 20, this will be 27. So extra workers are 27. Okay, then coming to next question. So these basically are all questions are of maths. Okay, part A. Now let's do some science-based questions. So this is part B. So in part B, second question, the key driving force for membrane transport is membrane thickness, distance, molecular size, concentration gradient requires. So here, driving forces. So driving forces are gradients in chemical potential, electrical potential, hydrostatic pressure. Okay, so membrane transport, that is through active and passive. Okay, so here the answer is concentration gradient. So this is by active and passive means. So basically driving forces are chemical potential, electrical potential, then hydrostatic pressure. Okay, coming to the third question, dash enzyme catalyzes conversion of fructose to fructose 6 phosphate, requires dash as cofactor. Okay, third question, so this is from glycolysis. So in glycolysis, you might be knowing the conversion, right? So first step is that from glucose it converts to glucose 6-phosphate. It converts to glucose 6-phosphate, and that is through enzyme hexose kinase. Now from glucose 6-phosphate it converts to fructose 6-phosphate, and that is with the help of enzyme phosphoglucose isomerase. So now fructose 6-phosphate now gets converted to fructose 1,6-biphosphate, and that is with the help of enzyme phosphofructokinase. Okay, so now we need to find out from fructose to fructose 6-phosphate. Here we can only see fructose 6-phosphate, right? So first option is phosphofructokinase 1 Mg2 positive. So this enzyme is responsible for the conversion of fructose 1,6-biphosphate. Now phosphofructokinase 2. Now this is manganese 2 positive, Mn2 positive. So this is responsible for fructose 2,6-biphosphate. Okay. So now coming to option number 3, that is the answer, phosphofructokinase Mg2 positive. So firstly we know Mg2 positive, that is the cofactor. So Mn2 positive will automatically get cancelled. So Mg2 positive. So since 1 is there, so that is for this is for 1,6-biphosphate, this is for 2,6-biphosphate. So we only need to convert fructose 6-phosphate, nothing is there, neither 1 nor 2. So third option. Then next is pervaporation. So pervaporation is ion removal, separation of liquids, separation of solids, or water purification. So pervaporation, it's a membrane separation process in which upstream side of membrane is in contact with feed liquid while a vacuum is applied on downstream side of membrane. This is just an idea. So basically pervaporation, this is partial vaporization through non-porous or porous membrane. So this is the option number 2, separation of liquids. Okay, then coming to next question number 5. Agrobacterium tumefaciens is used for transformation in plant tissue culture because both agrobacterium and plants have similar genetic makeup. Agrobacterium genomic DNA can transfer its genetic material to plants. Which genes of agrobacterium aid in tDNA transfer to plants? All of these. So fifth question, agrobacterium, that is here all options are correct. Now plants and agrobacterium, they have similar genetic makeup, that's why they are used in plant tissue culture. This is from Plant Biotech. So next is agrobacterium genomic DNA. That's why it's used in genetic engineering. Third option, vir genes. So here vir genes are vir A and vir G. This is vir. Okay, so option number 4, all of these. Okay, then question number 6. Proteins responsible for structural maintenance of chromosomes are condensin, cohesin, collagen, both 1 and 2. So proteins, here we are talking about chromosomes. So if we don't know exactly, we might have an idea, like in chromosomes what we have read about, right? Which proteins, which terms we have read about chromosomes. So here if we talk about collagen, so collagen is most abundant protein in the body. It's like fiber, like it has fiber like structure, right? It is used to make connective tissue. So it is used to support or strengthen your skin, muscle, bones and connective tissues, right? So collagen, that is basically we have read it in a separate chapter, as a complete chapter about collagen synthesis. So collagen is a protein that is made up of amino acids. It provides structural support. So we haven't read anywhere in chromosomes, right? So this makes it easy. So collagen is eliminated. Collagen. Okay, next coming to condensin and cohesin. Cohesin, we all know, in chromosomes we have read about cohesins. Now condensins are SMC2 and SMC4. So cohesin and condensins are there. So this is both 1 and 2. Okay, then next question is question number 7. The number of a nuclear pore in a cell depends on the size of cell, transcriptional activity of cell, DNA content of cell, none of these. Number of nuclear pore in a cell, it depends on transcriptional activity of the cell. So transcription process, transcription, that is formation of RNA from DNA. Okay, now coming to next question. Optical density is based on light refraction, light absorption, light diffraction, light scattering. So this is the answer is light refraction. Lowest yield of ATP is achieved in anaerobic respiration, aerobic, fermentation, both 1 and 2. So, and lowest yield of ATP, that is in fermentation. Severe acute respiratory syndrome, coronavirus 2, is caused by coronavirus 2. They're talking about the structure of coronavirus, that is a single-stranded RNA virus. Now coming to 11, purity of DNA samples can be checked by measuring the ratio of absorbances at 260 and 280 nanometer. For pure DNA sample, the ratio should be. Question number 11. So now for pure DNA sample. Now for DNA, ratio is between 1.7 to 2.0. For RNA, it is 2.0 to 2.2. Now equivalent to 18, that is equivalent to 1.8, that is pure. Now less than 1.6, that means there is presence of proteins, phenols, contaminants. And so here we, now coming to ratio, that is 260 over 280. Then we have 260 over 280. Okay, so pure DNA sample, this should be 7.8. First option, this is like this 1.8. So if we see this option, this second option, it states less than 1.8. that means less than 1.8, that means it's somewhat impurities are there. Then proteins, phenols, so some impurities will be there if it's less than 1.0. that is less than 1.0, that is irrelevant. Obviously some impurities will be there. Then more than 2, that means more than 2, so 2 to 2.2, that is about RNA. So more than 2, that means degradation of RNA. So this is equal to 1.8. Next is key protein involved in complement activation is C5, C9, C8, C3. So this is about in chapter, from chapter complement activation, the answer is C3. 2-keto 3-deoxy-6-phosphogluconate is an intermediate of pentose phosphate pathway, enter-Dodorov pathway, Embden-Meyerhof pathway, tricarboxylic acid cycle. So this product here, they all are talking about glycolysis. So these all are the other names of glycolysis. EMP pathway, that is Embden-Meyerhof pathway, glycolysis or EMP. Okay, then pentose phosphate pathway. So here in this pentose phosphate, this is also glycolysis. Here NADPH term is there. So pentose phosphate, that is also called phosphogluconate pathway. Phosphogluconate pathway, or another name is hexose monophosphate shunt. That is HMP. So if we see pentose phosphate, that means NADH term should be there. If in question somewhere NADPH they are asking, that will be about pentose phosphate. Then enter-Dodorov pathway. So here in second option, in this pathway, these are all somewhere subparts of glycolysis. This 2-keto 3-deoxy-6-phosphogluconate is found in enter-Dodorov pathway. Krebs cycle, TCA cycle, that is Krebs cycle. So we are not talking about Krebs cycle, right? Okay, 14. The organelle absent in plant cells is vacuole, lysosome, chloroplast, centriole. So lysosome is everywhere, right? So lysosome, if we talk about plant cells, chloroplast will also be there. Lysosomes generally they are present everywhere, right? Vacuoles are also there. Centriole is not there. So this is option D. Now coming to 15, 70S ribosome is made up of 50 and 30S subunits, 50 and 40S subunits, 60 and 30S subunits, 60 and 40S subunits. So ribosomes, we have two subunits. One is 70S, other is 80S. One is large subunit, other is small subunits. In 70S we have 50S, 30S. In 80S we have 60S and 40S. 70S is 50 and 30. So if you see fourth option, here they are talking about 80S ribosomes. Okay, next enzymes for 16th question. Enzymes involved in RNA interference are dicer, cohesin, bicoid, maskin. Okay, dicer in RNA, this is topic from molecular biology, RNA interference. So dicer, along with dicer there is one more enzyme, that is drosha. Okay, so here dicer, that is this, here we have, this is basically RNase 2 enzyme. Okay, now cohesin, this is related to chromosomes, right? Maskin, this is rare term, we don't use this term mostly. So cohesin, that is related to that chromosomes, right? And bicoid you might have read in Drosophila. Bicoid gene is there, then along with that one more is there, Antinapedia. Then okay, so bicoid term is with that developmental biology. So here the that is 16th is first option, dicer. Okay, next, which one of the following is not true for euchromatin? 17th. Okay, one sec. Okay, then 17th question, that is not clearly visible. Okay, so 17, which one of the following is not true for euchromatin? Which is not true for euchromatin? Okay, so option A, they are transcriptionally active. Second option is they are less condensed regions of chromosomes. C is bar body is an example of euchromatin. D is all of these. So one term is heterochromatin and another term is euchromatin. So euchromatin, that is active, loosely packed, light stained, and less condensed. Okay, so they asking which is not true. Okay, transcriptionally active, this is true. This is less condensed, that is true. So they are saying bar body is an example of euchromatin. So see, bar body is inactive. So if it's inactive, it can't be an example of euchromatin. So this is not true, this is wrong. So answer is C. Next, 18th, glucose sensors utilizes as bioreceptor. Glucose oxidase, glucose kinase, glucose dehydrogenase, both 1 and 3. Utilizes as bioreceptor. This is glucose oxidase A. Then coming to question number 20. Resolving power of a microscope mainly depends on numerical aperture, refractive index, objective lens, ocular lens. So resolving power, that 1 minute. Resolving power, or you can say resolution. Resolving power or resolution. See, another question was optical density. Optical density was based on light refraction. Now here we are talking about resolving power. So this is from microscopy, resolving power. Okay, so the form resolving power, or you can say distance, small d, that is equal to 0.61 into lambda upon Na, or sometimes written as small n. Okay, so Na here is the numerical aperture. D is the distance, or you can say resolution or resolving power. Now resolution power is inversely proportional to lambda. And resolution power is directly proportional to small n. So RP is, this is another formula, 1 over d theta. N sine theta over lambda. Okay, so now here they are talking about resolving power. It depends on, see, objective lens, ocular lens, it is not there. Then wavelength is there and numerical aperture is there. So if somewhere wavelength they could have written wavelength, but so wavelength is also there and numerical aperture is also there, angle theta is also there. So here numerical aperture. If they talk about refractive index, refractive index, that is about reflection and refraction. Okay, then NRSE stands for notochord restrictive silencer element, neural ectodermal specification enhancer, neurorestrictive specification enhancer, neuron restrictive silencer element. So here let's start with N. N, here they are given notochord, neural, neuron. N is for neuron. R, restrictive ectodermal can't be there, restrictive, then all restrictive. Then S, silencer specification, silencer, its silencer. Then E for element or enhancer, E is for element. So notochord in any case is not there. We haven't read any abbreviation about notochord, right? Neuron, if you don't know about NRAC, but you in neuron chapter, that is related to brain, some full forms are there. Okay, so it's about neuron. Then is impermeability of mitochondrial inner membrane to most ion, electrons and protons is due to the presence of cardiolipin, collagen, chitin, cellulose. So collagen, that is related to your connective tissue for support. Okay, so collagen cannot be there. It is not for inner membrane for permeability or impermeability. Then, so if you know cardiolipin, that is a phospholipid. Cardiolipin is a phospholipid. Okay, so in membranes impermeability, phospholipids are there. That is cardiolipin. Then lateral root primordia developed from pericycle, endodermis, ectodermis, xylem. So this is from pericycle. Then Gram staining is based on the presence of phospholipid, peptidoglycan, lipid A, glycolipid. So Gram staining, that is based on the presence of peptidoglycan. Then for exogenic reactions, delta G or standard free energy change is negative and Ke is more than one. More than one means forward reaction. Then second option is delta G is positive and KEQ is more than 1, delta G is negative and KEQ is less than 1, delta G is positive and KEQ is less than 1. Less than 1 means reverse reaction. Okay, so here so delta G is equal to minus RT ln KEQ. So delta G is negative here. So the first option, delta G is negative and K equilibrium constant is more than 1 forward. Then we have next question, 26th. Ramachandran plot is first used for determination of stable protein structures, also called zero. This is also called phi plot. The C option provide information about sterically favorable conformations. D is all of these. Fourth is all of these. Used for determination of stable protein structures. Here Ramachandran plot, that is used for secondary secondary protein structures, then also called phi plot. So this phi plot, then we have another symbol psi plot, then provide information about sterically favorable conformation. So option number D, all of these. Then 27th, Michaelis, this is from enzyme and kinetics of enzyme chapter. Michaelis Menten constant Km is related to the enzyme's affinity for the substrate, enzyme's activity for the substrate, enzyme specificity for the substrate, enzyme selectivity for the substrate. So you can make judgment from the name of the chapter only, enzyme and kinetics, right? So if you see the formula, Vmax is equal to K2 into E naught, or sometimes referred to as K cat. Okay, so this is related with enzyme's affinity for the substrate. 28th, antibacterial action of penicillin could be attributed to inhibition of enzyme transpeptidase, inhibition of cross-linking of peptidoglycan side chain, hydrolysis of peptidoglycan, both 1 and 2. Inhibition of enzyme transpeptidase, so this is both 1 and 2. Which of the 29th, which of the following is not a pattern of cell movement during gastrulation? Ingression, epiboly, invasion, invagination. So here in gastrulation, we have the sequence as invagination, then involution, ingression, delamination, epiboly. So ingression is the third step, epiboly is the fifth step, invasion is not there, invagination is the first step, so invasion is not there. 30th, which of the following is not a possible catabolic fate of pyruvate? Lactic acid fermentation, citric acid cycle, ethanol fermentation, then glycolysis. Fate of pyruvate. So this is somewhat anaerobic respiration, right? Because alcohol, or you can say ethanol formation is there. So ethanol fermentation and lactic acid fermentation. If you take an example of athletes, right, and they do very too much exercise, right? So they are so in athletic people, this kind of formation of this kind of fate of pyruvate is there, like from anaerobic. because their heartbeat increases when the exercise is fast. So ethanol fermentation and lactic acid fermentation both are anaerobic. Then citric acid cycle, that is, this is Krebs cycle. So Krebs cycle, it starts with acetyl coenzyme A. And acetyl coenzyme A, it comes under aerobic. So if you know the sequence, first we have glycolysis, after that Krebs cycle, that is here we have CTA cycle, sorry TCA, tricarboxylic acid, TCA or citric acid cycle. After Krebs cycle, then we have electron transport chain or system, right? So this is glycolysis. So this is not a possible catabolic fate. It's a fourth option. Okay, now 32nd question. 32. Which one of the following is the correct sequence of prophase 1 stage? Option A, leptotene, packetine, zygotin, diplotene, dikinases. Option 2, leptotene, zygotin, packetine, dikinases, diplotene. Third, zygotin, leptotene, packetine, diplotene, dikinases. Fourth, leptotene, zygotin, packetine, diplotene, dikinases. So coming to prophase 1 stage, first it is leptotene. Here we can see centromere. Then we have zygotine. Here we see synaptonemal complex. And then third, it's packetine. Here we have the formation of tetrad, that is crossing over takes place. Then we have diplotene. Formation of chiasma. Chiasma is formed at the point of crossing over. Then at last we have dikinases. Okay, so option number 4. Then 33rd, separation of components in gas chromatography is based on molecular weight, partition chromatography, molarity, conductivity. Here they're talking about gas chromatography. So molecular weight is not there. Molarity, this is about molarity, that is in chemistry somewhere. Cis partition coefficient. Then 34th, according to Bohr effect, the oxygen transportability of hemoglobin is more when pH decreases, pH increases, pH remains stable, it is not affected by pH. So in this is a chemistry based question. So in Bohr effect, more oxygen is released if more oxygen is released, it weakens the hemoglobin and oxygen bond. So if bond is getting weakened, so here pH is decreasing. Okay, next 35th, which of the following is not associated with the extracellular matrix of the cells? Extracellular matrix, that is ECM. So proteoglycans, collagen, cellulose, fibronectin are not associated. Collagen, integrin, it is there. Fibronectin, proteoglycan is there. Cellulose, that is associated with plant cell wall. Then is CRIS. PR is full form is, it's clustered, then regularly or restrictive, regularly, then interspaced, short palindromic, repeats. Clustered, regularly interspaced, short palindromic, repeats. 37 dextrans are polymeric glucose, polymeric fructose, polymeric galactose, polymeric lactose. Dextrans are polymeric glucose. Next 38, which of the following is not a type of microtubule associated with mitotic spindle? So here we have three, one sec. Here we have three types of microtubules. Kinetochore, polar microtubules, then astral microtubules. So central is not there, so this is not a type of microtubule. 39, the enzyme responsible for removal of DNA supercoils is helicase, DNA ligase, DNA polymerase. Ligase is for joining, helicase is for unwinding. Okay, so DNA polymerase, that is for replication. So topoisomerase, this is a type 2 enzyme. Then 40, diethyl amino methyl DEAE. Cellulose is an anion exchanger. Anion is negative. It will prefer to bind with proteins having no charge, positive charge, negative charge. Binding does not relate to charge. This is question from this is chromatography from cation and anion exchange. This is from so this is about anion. So if anion is negative, it will attract to positive charge. 41, which of the following is not a part of endomembrane system? So endomembrane system, it consists of nuclear envelope, then endoplasmic reticulum, then plasma membrane. So here we have Golgi body, lysosome, vesicles. Golgi body, lysosome and vesicles. Okay, ribosome is not there. Next, 42, unit of maximum specific growth rate, mu max is, this is pronounced as mu, mu, unit, right? Maximum specific growth rate, that is basically minute inverse, or you can say hours inverse, because since minute is not there. Then 43rd, phenylketonuria is caused by dysfunction of phenylalanine hydroxylase, phenylalanine monooxygenase, phenylalanine mutase, phenylalanine dioxygenase. This is from chapter health, human health and diseases. Phenylketonuria, that is caused by the dysfunction of this phenylalanine hydroxylase. Okay, then 44th question. Which of the following electronic transitions is not possible in UV visible region? Which of the following electronic transitions is not possible in UV visible region? This is from biochemistry. N to pi star, you might have read about bonding and anti-bonding electrons, right? If I talk about simple simple sigma, this is bonding. Sigma star means non-bond, anti-bonding. Pi means bonding, pi star means anti-bonding. N to pi star, then pi to pi star, sigma to sigma star, none of these. N to pi star, this is in N to pi star, this is in C double bond O. Then sigma to sigma star, this is star is there. Sigma star, this is CC. Then pi to pi star, that is in C double bond C. Basically this is none of these. These kind of questions generally don't come. Mostly this is chemistry based, like this is for biochemistry. Okay, next. Uh, 45th, product of dissimilatory nitrate reduction is ammonia, nitrogen, nitrate, nitric oxide. 45th. So first we have two terms, NO, then NO2, then N2O, and single N2. Single N2, you all know, nitrogen. Okay, so here now we have single NO, that is, this is nitric oxide. When one nitrogen and one oxygen is there, that is nitric oxide. Now coming to N, now two oxygens are there, NO2, that is nitrite. Okay, when NO2, we talk about NO2, that means nitrite. Okay, so nitrite, so because two oxygen are there, so this is a dioxide. Okay, then there is another, that is NO3. So now NO3, that is nitrate. So this is simple, only one is nitric IC. With 2 we have ite, with 3 we have 8. These two are important. Then N2O, that is nitrous oxide. So here they are talking about a nitrate reduction, that means NO3. So in nitrification we have NO3 negative. Okay, so here ammonia, that is NH3. Then nitrogen is N2, nitrate, that means they are talking about NO3. Nitric oxide, that means they're talking about NO. So this is from nitrification and denitrification. What happens? NO gets converted to N2O, N2O further gets converted to NO, then N2O, then N2. So this is a process, and at the end ammonia is there, ammonia is formed. Okay, then 46th, formation of glycogen cannot be initiated in the absence of enzyme. Glucokinase, glycogen phosphorylase, glycogen kinase. So this is from glycogenesis topic. So we just need to see which enzymes are there. So all are there except glycogen phosphorylase. Formation of glycogen cannot be initiated cannot be initiated in the absence of enzyme. Means which enzyme is important, which if we see glycogenesis process, this enzyme is there. That means glycogen phosphorylase is there, that is that should be present. Okay, 47, histones are rich in lysine and arginine, lysine and asparagine, glycine and lysine, leucine and arginine. We just need to see basic amino acids. That is lysine and arginine, and they both are positive. RK, their there another abbreviated name, arginine and arginine R and K lysine. Then 48 match list 1 with list 2. Translation occurs in cytoplasm, okay, and transcription occurs in nucleus. Translation, formation of amino acids, proteins, right, amino acids, they all take place in cytoplasm. And if we talk about transcription, that is in nucleus, that is RNA. Transcription nucleus and translation cytoplasm. Replication, that is in nucleus. ATP synthesis, that is in mitochondria. Dark reaction, that is about stroma. And light is in thylakoids, grana. Okay, so option number 4. 49th, intramolecular rearrangements are catalyzed by transferases, oxidoreductases, isomerases, lyases. So if you don't have an idea about this question, so you can simply see from this word, they are talking about rearrangement. Okay, rearrangement means number of molecules are just same, they are just number of molecules, they are just changing its scenario, right? Only isomerase can do that. Then 50th, amphibian eggs undergo equal radial holoblastic cleavage, unequal radial holoblastic cleavage, equal radial meroblastic cleavage, and equal radial meroblastic cleavage. Equal radial holoblastic cleavage, that is symmetrical. Which substance determines the type of cleavage during development? Nucleus, cytoplasm, yolk, both 1 and 2. This is nucleus as well as cytoplasm, both 1 and 2. This is from developmental biology, in case of sea urchin. Okay, then 52nd, protein purification in a two-dimensional gel electrophoresis is based on molecular weight, number of subunits, charge, both 1 and 2. Okay, so protein purification in a two-dimensional gel. So in one-dimensional we have isoelectric point, in second dimensional we have molecular weight. So if we see isoelectric point, that is all about pH, high pH and low pH. Here we have high pH, here we have low pH, negative, here positive. So here this is both 1 and 2, molecular weight and charge. Why I am saying molecular weight? Because in SDS page here that principle of molecular weight is used. SDS page electrophoresis. Okay, 53rd, ATP molecules produced by oxidation of one molecule of palmitoyl coenzyme A2, CO2 and H2O are. This is 53rd question. ATP molecules produced by oxidation of one molecule of palmitoyl coenzyme A to CO2 and H2O are option A 38, option B 108, C 148, D 40. So this is A 38. Coming to question number 54. Which of the following regulation occurs in slowest time frame? Feedback regulation, allosteric regulation, regulation through covalent modification, enzyme synthesis through gene induction. This is 4, enzyme synthesis through gene induction. Then 55. Which of the following sentences is not true for pyrosequencing? It is based on detection of pulses of chemiluminescence, requires the activity of enzyme sulfurylase, requires addition of dideoxynucleotides, none of these. So pyrosequencing, basically it's about pyrophosphate release. Pyrophosphate release, right? So even if you don't know about pyrosequencing, but you might know about Sanger sequencing, right? So in Sanger sequencing, we all know that dideoxy DNTPs are used. DDNTPs, right? So if Sanger sequencing includes DDNTPs, it can't be in pyrosequencing, right? So option number 3. So here they are asking which is not true. 56. Which of the following proteins is associated with amphibian development? Collagen, fibrin, catenin, keratin. Collagen, that is related with connective tissue. Then fibrin is not there. Coming to catenin, if this is for, let's say for example, EP cadherin. Catenin, that is if you know about Wnt signaling. Okay, so Wnt catenin, that is in frog embryo. So option number 2. 57. For each ATP molecule breakdown, the sodium potassium pump causes movement of more Na positive out and less K positive in, K positive out and Na positive on one basis, Na positive out and K positive on basis, K positive and Na positive in same direction. So 57th, sodium potassium pump. Now what happens in sodium potassium pump? Here we have plus sign, that is Na positive. Here we have K positive, Na positive. Here we have K positive. So these Na positive are outside. Movement is like this, here to here. So Na positive, they are moved out, and less K positive, they are inside. K positive, 3 sodium exit, and 2 K positive. 2 K positive enter. Sodium out and potassium in. So this is not on one-on-one basis, this is on 3, this is on 3 and 2 bases, right? 3 for sodium and 2 for K positive. So more Na positive, option number 1, more Na positive out and less K positive in. Okay, then coming to 59th question. Which of the following is not a secondary messenger? Ca2 for calcium. 1 minute. secondary messenger. This is from cell signaling, GPCR, G protein couple receptor. So Ca2 positive, this is a secondary messenger. CAMP is a secondary messenger. Then IP3 is a secondary messenger. DAG, diacylglycerol, is a secondary messenger. NO is a secondary messenger. CGMP is a secondary messenger. Right, so calcium, CAMP, DAG, diacylglycerol, that is there. Now AMP, if we talk about AMP, that is adenosine monophosphate. This is basically adenosine monophosphate. Then if we talk about ADP, that is adenosine diphosphate. ATP, adenosine triphosphate. So all these things that doesn't come in secondary messenger. Okay, so option number 4, this is not a secondary messenger. Okay, then 60th, no, not 60th. Then question number 61. Pectin is a polymer of glucose, glucuronic acid, galacturonic acid, or idiuronic acid. So pectin, that is related with plant cell wall. So in plant cell wall, it includes cellulose. Whenever you see the word cellulose, that means they are talking about plant cell wall, plant biotechnology. Then it includes hemicellulose, then lignin. So pectin is a polymer of galacturonic acid. Then in S62, an enzyme without its required cofactor prosthetic group is referred as coenzyme, apoenzyme, holoenzyme, catalyst. Now cofactor, now it is further divided into inorganic cofactor, organic cofactor, then metallo-organic. Metallo-organic. So inorganic, that means this is metals. Inorganic, we have metallo enzyme, then we have metallo-label enzyme, organic, then organic comes out to be coenzyme and prosthetic group. Okay, then third one is as it is, metal-organic. One minute. So this one, metal activated enzyme. Okay, now organic we have coenzyme and prosthetic, right? So here we have a term holoenzyme. So in holoenzyme means apoenzyme plus cofactor. So now here we have apoenzyme. If I say apoenzyme or proenzyme or zymogen, same thing. Apoenzyme, that is inactive. Okay, so in without cofactor, that is inactive. So in enzyme without its required cofactor prosthetic group, that is called apoenzyme. Okay, then we have 63rd, triple helical coil structure is characteristic of. Collagen is triple helix. Now cellulose, that means they are talking about plant cell wall. Chymotrypsin can't be there, elastin is not there. Collagen is triple helical structure. This is the first line in chapter of collagen. 64th, mammals do not have ability to convert fatty acids to glucose because they cannot undergo glycoxylate cycle. There is no need for such conversion. They can obtain glucose from other pathways, both 1 and 3. Now 65, allosterically regulated enzymes are. So basically allosterically means an extra site is there which can change shape. Like two sites will be there, extra site. One extra is there, so two sites can be there. So their oligomeric proteins possess regulatory and catalytic domains, shows sigmoidal behavior. Sigmoidal behavior means like this, S shaped. This is S. This is sigmoidal curve, S shape. So it allosterically regulated enzyme. Here what happens, it changes its shape. Possessed domains show sigmoidal behavior, both 2 and 3. 66, difference between chromatin regulation by high and low CpG promoters is. High CpG content of promoters, this is from DNA methylation. Repressed gene, high CpG content promoters repressed gene by DNA methylation. Now another we will see same, high CpG promoters repressed by histone methylation. Histone methylation is nothing, we have read about DNA methylation. Okay, and then second option, low CpG content repressed gene by histone. Again histone word is there. Low CP. Now fourth option, low CpG content promoters repressed gene by histone acetylation. This is somewhat in transcription. So you can make an idea from this that this if we talk about CpG, right, this CpG we have read in DNA methylation. Okay, now 67th, in reverse phase liquid chromatography, stationary phases. This is question from chromatography, like types of chromatography. There gel filtration chromatography is there, column chromatography is there. Okay, this is also a type of chromatography, reverse phase liquid. Here what happens, there stationary phase and mobile phase. Stationary phase is non-polar. Non-stationary phase is non-polar, and mobile phase is polar. 68, lipopolysaccharide present in gram-negative bacterial cell wall is considered as an endotoxin because of lipid A, core polysaccharide, O sites and integral proteins. If there is confusion between 1 and 3, right? So O site, if we talk about O side chain, this is component of LPS. Component of LPS. So it is an endotoxin because of lipid A. Coming to 69th question. Sedimentation rate of a particle will be 0 when density of the particle is less than surrounding medium. Second option is density of the particle is more than surrounding medium, is more than the surrounding medium. Third is density of particle and surrounding medium are equal. Fourth is density of particle is twice as the surrounding medium. So it's 0 when they are equal. Now coming to 71, activity of protein kinase A is regulated by So 71, so this is from GPCR signaling. Now what happens, GPCR, here we have G alpha S, I mean S for stimulatory, or you just simply write it G alpha. Okay, then we have adenylyl cyclase. It further activates CAMP. Okay, now CAMP helps in activation of PDE phosphodiesterase to AMP. Now CAMP, it further activates pKa, 4 units, pKa, that is protein kinase A. So further it then we have Krebs here. This is kind of GPCR signaling. Now here they are asking about activity of protein kinase A is regulated by, if we see we have CAMP, right, but in option CAMP is not there, so adenylyl cyclase. And this phosphorylase kinase, this is responsible for glycogen breakdown. Now question number 73, which of the following is a molecular motor? So molecular motors, we have dynein, kinesin, myosin, ATP synthase, flagellum motor, so here we have kinesin. Then we have 75. Cells are small in size because it provides more surface area to volume ratio. It provides less surface area to volume ratio, provides better both 1 and 2. So it is less small sizes, less, so more surface area to more ratio. Size is small and ratio is more, inversely related. Okay, so these all were MCQs about for M.Sc and Ph.D entrance examinations. Okay, so this was about it. Thank you.